Okay, I want to start today's deep dive with a little challenge, a thought experiment for you.
All right, I'm in.
I want you to just picture your body fat. You know, the stuff on your stomach or your thighs, the stuff we're all sort of trying to get rid of. If I asked you, what is that fat doing right now? What would you say?
Well, most people would say it's doing nothing. It's just sitting there, like a backpack full of energy you're not using.
Exactly. Dead weight, passive storage. But what if that's completely wrong? What if I told you that your fat is actually an active, intelligent organ?
Yeah.
That it's constantly talking to your blood vessels in this like secret molecular language.
And that conversation is what determines whether your arteries stay healthy and flexible or if they start to get stiff and, well, fibrotic.
That is the mystery we are unpacking today. We are looking at a really groundbreaking paper. It's titled Ablation of Prdm16 and Beige Fat Identity Causes Vascular Remodeling and Elevated Blood Pressure.
It reads less like a biology paper and more like a detective story, really.
It does. We have the crime, high blood pressure. We have a suspect, this, confused fat cell. And we find this villain, a rogue enzyme that's basically molecular superglue.
It's a fascinating mechanism. I mean, it really challenges how we think about hypertension. It's not just about salt or stress.
Yeah.
It's about the identity of your fat.
So let's set the stage.
Right.
To understand the crime, we have to meet the players. The paper talks about 3 kinds of fat: white, brown, and beige. Now, I know white fat.
Right. We all do. That's the classic energy storage unit.
Hoarder.
The hoarder, exactly. Under a microscope, it's called unilocular, which just means it has one big droplet of lipid inside. Its whole job is to save energy for later.
Okay, so that's white fat. Then you've got brown fat, or BET.
Yeah, and brown fat is the total opposite. It's multilocular, so lots of tiny droplets, and it is just jam-packed with mitochondria.
The cell's power plants.
Right. So its job isn't to store energy, it's to burn it, to generate heat. That's actually why babies don't shiver much. They have a lot of brown fat keeping them warm.
So, white stores, brown burns. Where does beige fat fit in?
Beige fat is the really interesting one for us adults. It's a hybrid. It actually lives inside your white fat, normally just kind of hangs out, but it's inducible.
Inducible, meaning you can switch it on.
Exactly. You expose yourself to something like cold, and it wakes up. It starts acting just like brown fat, burning energy. It's a shapeshifter.
And the person in charge of this whole shape-shifting operation is a gene, right? Prdm16.
Well, correct. Prdm16 is the transcriptional co-regulator. I mean, think of it like the manager on a job site. As long as Prdm16 is there, it's telling the cell, you're beige fat, your job is to burn energy.
Okay, so in this study, the researchers, they fired the manager.
They did. They created what's called a knockout mouse. They deleted the Prdm16 gene, but only in the fat cells. Nowhere else.
So the mouse is fine, except its fat cells have developed total amnesia.
That is a perfect way to put it. The beige fat completely forgot who it was. The study showed that it's thermogenic markers, like a protein called UCP1 that makes the heat just plummeted.
The fat went on strike.
It was worse than that. It actually switched sides. It started turning on genes you only see in white fat. It basically reverted to being a lazy storage unit. But here's the kicker. The real problem wasn't that the mouse got a little fatter.
It was what happened to the blood vessels right next door.
Exactly.
And we should be specific here because this isn't just any fat. We're talking about something called PV.
Perivascular adipose tissue. PVT. Your major blood vessels, like your aorta, they aren't just floating in your body. They're wrapped in this sleeve of fat.
So my aorta is wearing a fat jacket.
It is. And normally that's a good thing. That jacket is made of healthy, beige-like fat that protects the vessel. But in these knockout mice where the fat had its identity crisis, the vessel underneath started to change.
They call it vascular remodeling.
Which is a very polite way of saying the vessel got destroyed.
Right.
It became fibrotic. The researchers found this huge increase in collagen and fibronectin in the vessel walls.
Let's pause on fibrotic. What does that actually feel like for a blood vessel?
Okay, so think of a healthy artery as a new rubber band. It's flexible, elastic. When your heart pumps, the artery stretches to absorb the pressure, then it snaps back.
That keeps your blood pressure stable.
Right. Fibrosis is like pouring concrete over that rubber band. The vessel wall gets stiff, rigid. It can't stretch anymore.
So the heart has to work way harder to push blood through this stiff pipe.
Precisely, and that's what creates high blood pressure. But it wasn't just stiffness. The vessels were also hypercontractile.
Meaning what?
When they tested them with angiotensin II, a hormone that makes vessels tighten, The vessels from these mice just clamped down way too hard, aggressively.
So they were stiff and they were twitchy. They overreacted to stress.
You got it. And that led to systemic hypertension. The mice had high blood pressure, both systolic and diastolic, even though their body weight hadn't really changed. The damage was coming directly from the fat's personality change.
Okay, this is a lot to take in. We're about 5 minutes in, so let's do a quick recap to make sure we've got this straight before we dive into the how.
Good idea. So here's where we are.
Point 1.
We have beige fat, which is the good energy-burning fat.
Point 2, a gene called Prdm16 is the manager that keeps it beige.
Right. Point 3. Researchers delete that gene in mice, and the fat forgets its job. It turns into dysfunctional white fat.
And this is happening in the PVAT, the fat sleeve around the blood vessels.
Yes. And as a result, point 5. The vessels get stiff and overreactive, which causes high blood pressure.
So we have the crime scene, the stiff artery, and the suspect, the amnesiac fat cell. But we don't have the weapon. How does a fat cell sitting outside the vessel make the wall inside turn to concrete? Is it like physically squeezing it?
That was the million-dollar question. Is this mechanical or is it chemical? And to figure it out, they did this really clever experiment. It's called a conditioned media experiment.
I love this part. It's like the poison in the well test.
It is. So they took the bad fat cells, the ones without Prdm16, and they grew them in a dish with this liquid nutrient broth. They just let the fat cells sit there for a while, secreting whatever it is they secrete.
So the broth basically becomes a tea made of whatever the fat is leaking.
A toxic tea, it turns out. Then they took the fat cells out completely. They just took that liquid, that tea, and they poured it onto healthy, vascular, smooth muscle cells in a different dish.
So the cells from the artery wall never even saw the fat. They just touched the liquid it was swimming in.
Exactly. And the result was immediate. The healthy muscle cells started acting sick. They started turning on all these fibrotic genes.
That's the smoking gun. It's not physical pressure. The fat is sending a chemical message. It's mailing a letter to the artery that says, get stiff.
Precisely. It's a paracrine signal. Yeah. So the hunt was on for that specific molecule. They did RNA sequencing, looking for a gene that was, 1, regulated by Prdm16, and 2, that coded for a secreted protein.
They were looking for the author of that letter.
They were, and they found a few suspects. One was angiotensinogen, which kind of makes sense, but the levels weren't really up in the blood. It seemed to be a local problem. And then they found the real culprit, an enzyme that was massively upregulated. It's called QSOX1.
QSOX1. Quiescent sulfhydryl oxidase 1. Okay, what on earth is that?
This is the villain of our story. QSOX1 is an enzyme, and its job is to create something called disulfide bridges.
Okay, break that down for me. Disulfide bridge.
Imagine you have all these protein strings floating around in the space between your cells in the vessel wall. A disulfide bridge is like a chemical staple. It's a drop of superglue that permanently binds those strings together.
So it connects things. I mean, that sounds useful sometimes.
In moderation, yes. It gives tissues their structure. But the problem is this dysfunctional fat is pumping out huge amounts of QSOX1, so it floods the vessel wall and just starts stapling everything to everything else.
It's turning the flexible mesh of the artery into a solid block.
That is the molecular definition of fibrosis. But it does something else too. It also suppresses the vessel's natural antioxidative genes. So it's not just gluing the vessel shut, it's also preventing it from cleaning up damaging oxidative stress.
Which explains the twitchiness, the overreacting we saw earlier?
Exactly. So QSOX1 is this dual threat weapon. It causes stiffness through the disulfide bonds, and it causes hypercontractility through oxidative stress.
You know, there was a really interesting twist here with gender.
Yes, this is so important. They noticed that female mice were mostly protected from this. Even with Prdm16 knocked out, they didn't get the same severe fibrosis or high blood pressure.
Why not?
Well, it turns out that female mice naturally have lower levels of Prdm16 in that fat jacket to begin with. So when the researchers deleted the gene, the drop wasn't as dramatic.
Oh.
And because the system wasn't relying on it as much, the spike in our villain enzyme, QSOX1, was way smaller than in the males.
That's fascinating. So the male biology was uniquely vulnerable to this because it was so dependent on that one manager gene.
It really just highlights how important sex differences are in cardiovascular disease. The pathways are not always the same.
Okay, we're passing the 10-minute mark. Let's do another quick recap. We have officially unmasked the villain.
All right, here's the summary. Recap 1. The experiment with the toxic tea proved the fat was secreting something.
Recap 2. That something is an enzyme called QSOX1.
3. QSOX1 acts like a molecular superglue, creating disulfide bonds that make the vessels stiff.
And 4, it also boosts oxidative stress, which makes the vessels twitchy.
And finally, males get hit harder because of their baseline gene expression.
Okay, so this brings us to the climax. We have a theory. Prdm16 goes down, QSOX1 goes up, the vessel gets stiff. But you have to prove it. You have to show that if you stop the villain, you stop the crime.
And that is exactly what they did with the double knockout mouse. This is the ultimate test. They bred a mouse where they deleted Prdm16 so the fat is still confused. But they also deleted the gene for QSOX1.
So they fired the manager, but they also took away the superglue.
Perfectly put. And the results were, they were crystal clear. The fat tissue itself still looked white. It still had its amnesia.
Because Prdm16 was still gone.
Right. The fat was still broken. The blood vessels were perfectly healthy.
No way.
No fibrosis, no stiffness, no high blood pressure.
That's incredible. So you can have bad fat, but if you take away QSOX1, it can't actually do any damage to the blood vessel.
It proves that QSOX1 is the necessary mediator. It's the arrow. If you can break the arrow, it doesn't matter what the archer's doing, the target is safe.
This changes everything, but I have to ask the question everyone listening is thinking right now. This is fascinating, but I'm not a mouse. Does this pathway actually matter for humans?
The most important question, and yes, the paper brings in some really strong human data. They looked at a cohort of about 1,700 patients, and they split them up. People with detectable, active brown fat and people without.
And what did they see in the people who didn't have that active good fat?
They saw signs of what's called end organ damage. Specifically, their left ventricles, the main pumping chamber of the heart, had thicker walls.
Now, a thick heart wall sounds like a strong muscle. That's bad.
It's very bad. In the heart, thickness means the muscle is struggling. It's working against too much resistance, like constantly trying to push open a door that's stuck. Over time, that overworked muscle gets inefficient and fails. It's a classic sign of chronic high blood pressure.
So the people without the good fat had the same kind of heart stress as the mice with the broken fat.
Exactly. And they went even further. They looked at the UK Biobank, this huge genetic database, and they found that humans with certain genetic variants of the Prdm16 gene are statistically more likely to have hypertension.
That seals it. This beige fat-to-vessel health conversation isn't just a mouse thing. It's happening in us right now.
It is. It suggests that the quality of your fat, not just the quantity, is a huge predictor of your cardiovascular risk.
That really is a paradigm shift. We're so focused on BMI and weight. But this study says you could be thin but have angry, dysfunctional fat that's just pumping out QSOX1, and your arteries would still be in trouble.
Right. And on the flip side, you could have adipose tissue that's really metabolically active, really beige, and it's actively protecting your heart by keeping that QSOX1 in check.
So let's bring it all home. What's the so what here? How do we use this information?
I think there are two huge takeaways. The first is pharmacological. QSOX1 is an enzyme, and we're really good at making drugs that block enzymes.
An inhibitor, a glue dissolver.
You got it. If we can develop a specific drug to block QSOX1, we could potentially treat fibrosis and hypertension in a completely new way by stopping the stiffening at its source.
That would be a game changer. And the second takeaway
is lifestyle. We said beige fat is inducible, and we know things like cold exposure ice baths, even just turning down the thermostat, can activate it.
So when I'm shivering in a cold plunge, I'm basically telling my fat cells, hey, wake up, remember you're beige.
And by doing that, you are keeping Prdm16 active, which in turn suppresses QSOX1. You are literally telling your fat to put down the superglue.
I love that image. We have some control over this conversation. It's not just our genetic destiny.
Absolutely. The conversation between your fat and your vessels is happening 24/7, and you have the ability to influence what's being said.
This has been an incredible deep dive. We started with a single confused fat cell, and we ended up with a whole new way to think about heart health.
It just shows you how interconnected everything is. You can't treat the heart without thinking about the fat that's wrapped around it.
So before we go, I want to leave our listeners with one final thought. We spend a lot of time thinking about what we feed our bodies. But maybe we also need to think about what we tell our bodies. If your fat cells are listening, what is your lifestyle, your environment telling them to say?
And keep an eye on the news for QSOX1 inhibitors. You might have heard it here first.
Exactly. Thanks for diving deep with us today. We'll see you on the next one.